just to recap where we left off last week, we were just talking about general cell structures. And last week we talked in great detail the structure of the plasma membrane and molecules which you would find in there. It even kind of went in more detail about proteins associated with the cell membrane. Then we went on and talked about the structure of the nucleus in decent detail. Okay, maybe in more detail than you've identified before, maybe just called the nucleus. And so we talked about some structures of it. And then where we left off was looking at components that you would find in the cytoplasm or the intracellular fluid. We talked about inclusions and other components in there. And then we were leaving off with organelles, which are suspended in the cytoplasm or intracellular fluid as well. We just defined our organelles as membrane-bound structures. We gave that a limiting definition, so we're not just spending 3 days talking about what you might find inside of a given cell, which is, there are a lot of different molecules and structures which you would find inside of a cell. But we want to review the primary role of some of these membrane-bound organelles. The first one that we talked about on Thursday was the smooth ER. And we'll go back and revisit its structure and its function. Then we're going to talk about the rough endoplasmic reticulum, which ties us into its association with another organelle called the Golgi Complex. We'll also talk about some similar structures called lysosomes and peroxisomes. And we'll conclude our organelle discussion with the mitochondria. After we talk about the mitochondria, we're going to talk about other non-membrane bound structures, including a ribosome, which is clearly critical to cell survival, and the cytoskeleton. Which brings me to mention, reminds me, on your handout, it was brought to my attention last week. If you have it, if you could turn to it, your handout, number 1. On the first page, there's a list of these organelles that we're going to cover again. And all of our different cytoskeletal proteins, major classes are summarized here: microtubules, microfilaments, and intermediate filaments. And there's 3 listed. But there's really only 2 different types shown in the image. And so if you want to draw a third one in, that's okay. So if you want to draw a third one in the image and just label it as one of the others, it's okay. Pretty generic looking inside of this cell. Might catch that? So you can draw on the third type of cytoskeleton in there. Yes? All of these again? All of these Yes. Now all of those listed, everything, how many that is. All 12 pictures, all 12 items will be on it. Everybody catch what we're adding to the handout number 1? Any other questions? Okay, so let's go back and review the role of the smooth ER. What does it look like? Not flattened. The smooth ER is actually, it is kind of sacs, it is plasma membrane, but what? Not flat, more like tubes. Okay? The smooth ER is more tubular. The rough ER is more flat in description. The smooth ER is tubular, and what else? Where is it usually next to? Usually maybe adjacent to the rough ER, which is usually next to the what? The nucleus. So usually. What are some important roles of the smooth ER? Okay, so fat, lipids, steroids, what? Synthesis of those things, and it also is really important for storage of calcium. Very good, our cation. Okay. So let's go back and review that. I'm going to pull up this image for you. If we look at this image on the far right, the smooth ER is tucked back here in the far left-hand corner, where the tubes, okay, the blue tubes, you see the difference between the smooth ER versus the rough ER. The smooth ER, inside of its lumen, we used that word last week, are different enzymes and other molecules that help make things like our lipids that make up our phospholipids, or our cholesterol that we mentioned before. So that's an important role for the smooth ER. As well as, we'll probably spend more time with the smooth ER in the muscular section in the 3rd unit this semester in its role of compartmentalizing calcium. It stores calcium in there and it releases it. So it stores and releases calcium between the lumina of that smooth ER and the cytoplasm. And so it just regulates where calcium is. Is it free in the cytoplasm or stored in there? Does that make sense? In a flux. The word flux, okay? Between the two locations, based on what happens at the smooth ER. What questions do you have about the smooth ER at this point? Steroid synthesis, calcium storage are the primary roles that we'll see for this. So, we can also talk about the rough endoplasmic reticulum, okay, the rough ER. This one is more flattened, and what makes it characteristic is that it has ribosomes attached to it. We haven't talked about the ribosomes yet. However, we know that ribosomes are important for protein synthesis. And these two things, although they appear as a kind of bulleted items, really important that you understand the fate of the proteins that are manufactured at the rough ER. Does anybody know? If a protein's made at the rough ER, where is it probably going to go? If it gets incorporated with the nuclear envelope, maybe, but most are not. Okay, what else? Well, it will be processed in the Golgi, very good. And then what, after it gets processed in the Golgi, where might a protein manufactured at the rough ER, where's its ultimate fate of distribution? Where would a protein made at the rough ER ultimately go? To where? Maybe. So she said in the blood. So where? Outside of the cell. So proteins that are to be released outside of the cell that made it are made by that cell's what? Rough ER. Additionally, remember all those proteins that we drew on the board, that I drew on the board last week about channels, transporters, glycoproteins, etc.? Those were made where? In the rough ER. The rough ER, yes, it makes proteins, but it's proteins that will be incorporated that will be incorporated in the cell membrane, as I've listed here, item 2, or secreted out of the cell. A protein that's secreted out of the cell, like a hormone or enzyme, or albumin in the blood, whatever, if it's not kept inside of the cell, in the cytoplasm, it was manufactured by that cell's what? Rough ER. Additionally, those proteins that are associated with membranes get made where? In the rough ER. This is really important to know that if a protein is to be secreted, whether it's a hormone or just a transport protein or whatever, it was manufactured in some cells' rough endoplasmic reticulum. And so if we look at it, its membrane is usually connected with the nuclear envelope, not always, but usually, and it is flattened. And inside of there, that process, anybody know the process for making proteins, the name of it? It is a type of synthesis. What's the name of the term for protein synthesis? Starts with T. Translation. Translation can occur across the membrane of the rough ER, between the lumen of the rough ER, and it makes protein. If a protein is made at the rough ER, where does it first accumulate? Where do proteins made at the rough ER accumulate? When they're made, where are they getting dumped into? Where are they ending up once they're made? Like right in the rough ER. What part of where would you find the newly synthesized protein? The ribosome is used to make them. Okay, so where are the ribosomes? On the they're attached to the outside. The proteins accumulate on the inside, or we could use our new word. Well, the process is translation, but the proteins would accumulate what part of the rough ER? say it? Lumen? The lumen. Proteins will grow and elongate in the lumen of the rough ER. So they're actually kind of sequestered in the inside of the rough ER when they first get manufactured. Are they supposed to stay there though? No, they've got to go where? Eventually, maybe outside of the cell, okay, or associated with the membrane. And so what happens is we have to have the ability to take portions of the rough ER and pull it off from the rough ER. It will help form these little things called, I think I used the word on Thursday, vesicle, V-E-S-I-C-L-E, vesicle. The little vesicles will pinch off of the rough ER. So let's review. If a protein is manufactured in the rough ER, it will grow and elongate in which part of the rough ER? The lumen. Okay? Eventually, though, part of the rough ER with the little newly manufactured protein should do what? Pinch off and form a vesicle. Very good. The vesicle will be transported to our next organelle. What is the next organelle? The Golgi apparatus. You can call it the Golgi apparatus, the Golgi body, whatever you wish. But it's usually adjacent to the rough ER. I didn't realize this until last fall, but in most animal cells there's usually one Golgi apparatus, but there may be multiple of a Golgi apparatus. So we get like, for example, like plant cells, they may have 20 or more Golgi apparatus. So I always thought I just presumed there was always one. That's not the case. There may be multiple of these. And what is the job of the Golgi complex? What does it do? It gets the protein ready for transport. Okay. What? It repackages our newly made protein. And it processes them. And they already came in packaged. We've got to unpackage them and process them. What kind of processing can occur at the Golgi apparatus to a protein? What kind of processing could happen at the Golgi? What can we do to a protein? We've already synthesized it in the rough ER. Now we're going to process it in the Golgi. What can we do? Eventually, but first, before we ship it out, the question is, yes, it will be shipped out of the Golgi, you're right. But before we ship it, we've got to process it. What kind of processing can occur with or on or for a protein? Processing, modifying, what kind of modifications? Good. I drew some on the board last week, on Thursday. What kind of additions can I make to my protein? I may add other proteins, or more likely what could be added in here besides proteins. So think about our other molecules. Get on the right track. Lipids, or those are or what? Carbohydrates. Do you remember last week we talked about glycoproteins? Those sugars don't get added at the rough ER. They get added where? In the Golgi apparatus. Well, they get technically, that was wrong what I just said, but they get modified. The sugars get modified at the Golgi apparatus. So what the Golgi apparatus does is it receives vesicles. As we can kind of see here, the Golgi in the orange. The pink thing would be a vesicle that was originally part of which organelle? The rough ER. It gets pinched off and transported. It looks magical in this image. It's not magical. We'll come back to talk about how it happens very briefly. And it gets transported over the Golgi. So the vesicle membrane fuses with the Golgi membrane. And in the proteins, in the pink. Yes, but my whole life when I see yellow, I say pink. I don't know why. It's like one of those things. And I know that I'm saying it. So yellow and green are representing the proteins, okay? And the pink would be the vesicle. And so the little vesicle would fuse with the Golgi apparatus so that now these little proteins, the yellow and the green proteins, get dumped from the lumen of the vesicle to the lumen of the what? Golgi. Okay? And this Golgi apparatus, it looks static. We use that word. Can we use that word static? Static means what? We use it in the term of homeostasis, meaning what? Steady, right? It looks static, like it's not moving, but it's really cool. It's always treadmilling. Have you used that word before? You ever walked on a treadmill? So, although you are in place, right, the little device, whatever, the platform moves, the membranes of the Golgi kind of treadmill, if you will. So, it looks like, if you were to do a screenshot, it looked like it was stationary, but really the membrane's kind of processing through. Because we're always doing what to the membrane? We're always adding to it, processing, and always releasing. So it's always kind of changing. So let's go back. Proteins manufactured in the rough ER will get wrapped in a vesicle and then dumped, if you will, into the lumen of the Golgi. In the lumen of the Golgi, a very regulated process, but basically, if there's carbohydrate groups, all those proteins, they get modified to the right shape based on how it's coded for. So that when a protein is eventually released, it's considered to be mature, if you will. All the carbohydrate groups, sugar groups, whatever, are active. And so when it's ready to release, it's still right. Yes. In addition to just processing, it condenses the protein, so it actually segregates the proteins of the same type to make a cluster of the same type of protein. So instead of saying insulin and growth hormone and some membrane protein all jumbled together and released randomly, all of our insulin will be grouped together in a single vesicle. All of our growth hormone will be grouped together, and all of our little membrane proteins will be grouped together. And so you made a good note about the Golgi. Not only does it receive proteins, modify them, but it also, you can just use the word separate proteins, concentrate them. So all the same type of proteins are grouped together. And then eventually, they would be what? First released and then distributed. So they get built into a new vesicle. So the Golgi, it is also kind of flattened, if you will. It's freestanding from the rough ER, but it's definitely just working back and forth with the rough ER. And it also faces the cell membrane, because these proteins will eventually be what? Do what? After they've been modified, they'll be transported, distributed where? Outside of the cell, or in that cell's membrane. So it's facing the membrane because it's going to work with the membrane. So we identified already that it does receive proteins that are been sequestered in transport vesicles from the rough ER. We also identified it makes modifications, packages, and distributes these little proteins for secretion. The process is called exocytosis. I think it's chapter 4 that will really look at what exocytosis is. So I'll come back to that hopefully by Thursday. But basically, that vesicle will go and fuse with the cell membrane, kind of like what you can kind of see happens here at the Golgi. The plasma membrane join within the vesicle and the cell membrane so that the proteins are exposed to the extracellular fluid. Is that too brief, or does that make some sense? A little sense? Any questions? What is a vesicle? A vesicle is just a little membrane bound. proteins in it, okay, but it's just a little portion of phospholipid bilayer, and it's tiny. And what was added? Yeah, somebody add something? It's a little phospholipid bilayer. It's usually kind of ovalish, roundish in shape, and it's transporting things like proteins. In this case, definitely proteins. Could be transporting lipids, like we described the HDLs and etc. Maybe transporting our triglycerides or whatever. But it's just a way to ship, and they use the word cargo. That word kind of okay. The cargo is molecules, but it's a tiny little cellular structure that can be transported from one region to the other. Like we see here between the rough ER and the Golgi, and eventually the Golgi to the cell membrane. In this image, we can see some vesicles, like the two pink items, and these little orange structures. These little things right here. We'll use the term vesicle a whole heck of a lot this semester, and really beginning at the start of Unit 2. We'll see vesicles in the end of our nerve cells that release proteins that they're called neurotransmitters. So that's a good question to ask. Any other terms that I need to clarify before we move on? Next organelle. The rough ER and the Golgi apparatus work together to not only synthesize proteins, but to modify and package them for release from the cell. Is the rough ER the only place where proteins can be made? No. If proteins need to be made to function inside of the cell, should they be made inside of the rough ER? No. I will come back to that. Only the rough ER is used for making proteins that will be incorporated in the cell membrane or released from the cell. And the other protein that works inside of the cell, like if you looked in your handout, like a microtubule, okay, or if you remember from your DNA structure, you talked about histone proteins. Those kind of proteins are, you've used the word enzymes before. Or we talked about the peripheral proteins last week. If they're on the inside of the protein, or just inside of the cell, they get made differently. Alright, so the rough ER works with the Golgi complex. Alright. It does make other organelles like lysosomes. You can add in there, you can put comma, peroxisomes. Makes lysosomes and peroxisomes. Because what we're going to learn in just a moment, our lysosomes and our peroxisomes are just basically vesicles with enzymes or proteins inside of them. They'll eventually release their contents out of the cell. Here's an alternative view. We can see lots of different vesicles. There's certainly a receiving face, receiving side, and a shipping side. The side that receives is adjacent to the rough ER. The side that ships is adjacent to the plasma membrane. So on the side that receives from the rough ER, the proteins haven't been modified yet. And then by the time we get to the shipping side, if you will, they've been processed, concentrated, they're ready to be released at some point. Questions about the Golgi at this point? Alright. We just mentioned that the Golgi apparatus can also form other organelles inside of the cell, including lysosomes and peroxisomes. And so we'll see this similar structure of a vesicle with proteins in the inside of it. So if you notice, question is, what is it? It's a membrane, so it still has a phospholipid bilayer. There's going to be proteins and other structures that we identified incorporated in it. But what we really kind of focus on is what's inside. What's the cargo inside? Does anybody know what's inside of a lysosome? Complete your worksheet. vesicles, it is like a vesicle, so itself is a vesicle. So what is in there? Enzymes that are used to clean up like cellular debris, to digest things, to break down substances inside of the cell. And which macromolecule are enzymes? Enzymes are basically what? Proteins. So these proteins are functioning as specific enzymes. So once a lysosome is formed, what it has concentrated inside of it are enzymes called hydrolases. And note, how do you know this is an enzyme? This is the ASE, very good, the suffix. The ASE tells us these are enzymes. And they have something to do with water. The enzymes found in lysosomes carry out hydrolysis reactions. You've done that before, seen that word before. Hydrolysis reactions. So it uses water. Lysis does what? Break down. Okay. So these enzymes break down different types of molecules. Could be cellular debris, could be old organelles, could be a pathogen like a bacteria or something like that. The lysosomes can fuse with some foreign particle or damaged, right, broken down particle, and the enzymes inside of the lysosome would just immediately catabolize. What does that word mean, catabolize? Break down. Right? Bonds at a time. And as you guys already identified, the whole point is to get rid of foreign substances. I mentioned like pathogens, other cell debris, old organelles like mitochondria. They can't live forever. They've got to regenerate. And so our lysosomes can break those down as well. And sometimes it's referred to as the intracellular digestive system. Throughout the semester, you'll see me use these kind of air quotes, and those are the sarcastic, like, is an organelle a system? No. It's a tiny little structure within a cell. So, but the reason that term was coined is because why? Tell me why. It breaks down or it destroys substances. What questions do you have about the primary function of lysosomes? Fairly comfortable? There's a similar structure called peroxisomes that you may find inside of a cell. There are also membrane-bound vesicles with what inside of them? With what did you say? Enzymes. Which are basically proteins that got manufactured where? Modified in the Golgi but made in the rough ER. Eventually, before we move on to the peroxisomes, let me back up. We said products that are manufactured in the rough ER modified in the Golgi, they're going to eventually be where? In the membrane or outside of the cell? Here I just described something that's hanging around inside of the cell, breaking down stuff. After a lysosome does its work, what will it do? It'll purge its contents outside of the cell. So how long the vesicle hangs around in the cell just depends. But at some point, let's say a lysosome breaks down a bacteria, once it's done its work, it's going to eventually spew all that stuff, broken down stuff out. Which if you've ever had you can visualize if you've ever had an infection, think about a zit on your face or something like that, all that business, lysosomes purging all that stuff out. Right? So that's an example of something that would be cleared right off of, say, a white blood cell. Is that visual enough for you? It's outside of the cell. So the lysosomes, structurally, are like peroxisomes. They're vesicles that have proteins inside of them that work as enzymes. However, the peroxisomes are a little bit more complex in that they have not one, but two classes of enzymes inside of them. And these enzymes clearly are going to work together. These are compartmentalized together. We have oxidases and catalases. Again, we can still see the ASE suffix. Oxidases must has something to do with oxygen, but they also what is that prefix? Does that sound familiar? Oxidation, oxidizing, moving what? Electrons. Yeah. And so we'll definitely review that in chapter 3 about oxidation, reduction, etc. But we do have these enzymes inside of the peroxisomes, oxidases and hydrolases, that actually have something to do with oxygen and water. Excuse me, not hydrolases, catalases, excuse me. Oxidases and catalases are going to work together. Oxidases will oxidize or use oxygen to remove hydrogen from something called H2O2. What is that? Peroxide. Have you ever seen peroxide at work? It's real bubbly and reactive, right? Do you think bubbly and reactive is good to keep inside a cell? No. So what happens is when your body's exposed to like environmental toxins, even like alcohol that we drink, we know it's toxic to the body, to the cells. As you know, you've heard about people with liver disease from drinking too much, right? They're kind of overexerting their peroxisomes kind of can't keep them. Amongst a bunch of other things as well, the peroxisomes are working all the time. And what they do is catabolize products, and it produces something called peroxide. As soon as hydrogen peroxide is manufactured, it needs to be broken down to something that's not reactive and damaging. And so, as soon as hydrogen peroxide is manufactured, enzymes called catalases can use that peroxide to make what? Water and oxygen. Not toxic, and not reactive, very stable molecules. And so peroxisomes are also used to degrade potentially harmful, not necessarily whole pathogens, but chemicals that are in the environment, okay, that are hydrogen and oxygen rich. The oxidases will help break down the molecule to release peroxide. As soon as that's formed, what happens? Not yet. First, the hydrogen peroxide is made, and then what? The catalases can catabolize the peroxide into what? H2O and oxygen. Eventually, once the peroxisome is kind of exhausted, then it'll be released outside of the cell.
So the cells have the same thing.
Yep, and that's good. The question was, well, when the cell's done with something, then it gets rid of it. Yes, and that's why we have waste. Okay, that's why we actually have tangible waste that we have to get rid of, whether it's urine or whatever the case, or mucus that you have to cough up, or snot that you blow out of your nose, right, or zit that comes on your face, or whatever. The body's purging these toxic components. That's the organelle level first. Good question. Any other questions at this point? It's also involved in this process called beta oxidation, which historically in the last few semesters I've not had time to really talk about beta oxidation. So I'm going to kind of give a crash course just in case. Does anybody know what this is? Have you heard that word before? Sound familiar? Well, oxidation is removing what? Electrons. You may or may not use oxygen, but oxidation is removing electrons. So basically used to do what? So removing electrons, so what's happening to the molecule? Becoming what? It is becoming partially charged. And what are we doing to it? Right here at the bottom? Breaking it down. And so beta oxidation is breaking something down. Anybody know what's being metabolized in beta oxidation? I don't think so. Could be, but I don't think so. Your fats, lipids, fat catabolism. can be broken down this way as well. Identified a moment ago, the purpose of the peroxisome is to get rid of potentially toxic particles in the body. Whoops, excuse me. Any questions at this point about the smooth ER, the rough ER, the Golgi, Lysosomes, or Peroxisomes? Beta oxidation is a term to describe fat breakdown or fat metabolism. Additional questions? The next organelle that we're going to discuss is called the mitochondria. I'm going to spend a little bit more time on its structure because we're going to spend a whole heck of a lot of time on its function. All right, because what? Let's just throw it in there for today. Structure distinct. Very good. Before I kind of fill in the details about this mitochondria, well, I have it listed here. Okay, what does the mitochondria? What's an important role of mitochondria? To make something called ATP, which is a molecule for what? Energy transformation. Okay. So we're going to talk about mitochondria. We're going to talk about energy transformation in chapter 3. And then once we understand how ATP is made, we can use it through the rest of the semester. So we'll just say ATP, so you have a better appreciation of what it is and where it came from. It's the primary site of ATP synthesis. Is it the only site for ATP synthesis? Is it the only place we can make ATP in the mitochondria? No. Okay. So we're going to add in chapter 3, yes, we'll talk about the mitochondria, but also the cytokines as well. Let's look at the structure of the mitochondria. How many membranes? 2 membranes. So in your handout, or if you're using regular notes, I want you to draw a mitochondria. A single mitochondria, excuse me. An outer membrane. You can draw it kind of plain looking, if you will. In mitochondria in the mature form are described as kind of bean-shaped or kidney-shaped. Here it's real oval-shaped. But the outer membrane is a phospholipid bilayer. So what's in it? Wait, what? Fat. So yeah, like our fats and our phospholipids. What other type of molecules are in there? Proteins and sterols. I don't know if it's exactly cholesterol, but some type of sterol like cholesterol. Carbohydrates may be associated, branching out there as well. And so it's still complex, even though it's drawn boring right here in the image. So our phospholipid food mosaic properties apply to the outer membrane, as well as the inner membrane. But if you look at the inner membrane, what happens to the inner membrane? It's folded, right? And this is biology. We're in the logical world, so why is this folded? To have more surface area. So it can carry out a lot of reactions in this pretty small structure. So draw the outer membrane, draw the inner membrane and fold it. We're increasing surface area. It too is a phospholipid bilayer. It also has sterols and transport proteins, etc. within it. Keep this in perspective. This is surrounded by which fluid? The cytoplasm, the intracellular fluid, the cytosol, surrounded by the cell content and everything else. And notice that there's a little bit of space between the outer and the inner membrane. That space is not a vacuum like it looks in this image. It too has what inside of it? Some type of what? Fluid. And the fluid in that space, we'll talk about probably not today, hopefully tomorrow. But the region, does anybody know the name of the region in there? What could if I want to talk about just right here, what should I call that? The intermembrane space. So on your image that you just drew, label the area between the outer and the inner membrane. The intermembrane, I-N-T-E-R, membrane. I-N-T-E-R. Intermembrane space. Because that's the space what inter means between. Intermembrane space. We are without a doubt going to understand what happens at the intermembrane space. I need you to know where that is. So see the outer membrane, the intermembrane space, then the inner membrane, and then what's in the middle? The matrix. So label the matrix. And matrix is a generic word, so you would precede that with mitochondrial matrix. That's the mitochondrial matrix. What do you think is inside of the mitochondrial matrix? There's definitely fluid in there. Good. Fluid filled. What else? Adenosine triphosphate or ATP. And even what? Adenosine diphosphate. What else would we find in there? What do you think helps make that transformation? That we're going to study in chapter 3. What else do you think's in there? Is it just water and ATP? We can keep it generic. There's going to be enzymes in there. You remember the Krebs cycle? We'll study it coming up, but that's definitely enzymatically regulated, so all the byproducts. Tons of molecular reactions happening here, and intermediate products and structures as well. So it's densely concentrated molecules as well. In order to help make lots of what? ATP, right, and transform energetic molecules. Alright, so the outer membrane, then the what? Inter, make sure you throw that T in there. If you just say I-N-N-E-R, that inner means within, so that would be a completely different. Spelling-wise, it doesn't make a big deal, but anatomically it makes a huge deal. Okay, so we have that outer membrane, then the intermembrane space, then the inner membrane, then the matrix, mitochondrial matrix. On your image, why don't you go ahead and just draw some transporter proteins, some integral transporter proteins in each, the outer membrane and the inner membrane. Just draw some transporter proteins. Because we've got to transport products in and out. We're also going to use them to make ATP. I'm going to draw a big 4. You see the red on this image? You can just do that across the membrane if you'd like. And just, the inner membrane is just overwhelmed with these transporters. transporters to use. And let's see how ATP is kind of made. We're not going to go to the level that you do maybe in gym bio or cell biology, but we'll review it. What questions do you have on the basic anatomy of a mitochondria? All right, so we identified it's a primary site of ATP synthesis. We noticed that the matrix does contain enzymes and is going to regulate two primary processes called the citric acid cycle, or you may have called the Krebs cycle, and also the electron transport chain, or maybe you call the respiratory chain. But these are the two big processes that happen inside of a mitochondria. We also identified that it's not one, but two membranes around it. We also drew that the outer membrane is smooth, and the inner membrane folds, and the folds are called cristae. Can you see the labeling in this image? The inner membrane, the folds are called cristae. If we look at this statement, I'll explain this in just a second. The number of mitochondria varies within a given cell, which led me to this statement. The density of mitochondria in a cell indicates its E, what? What does that stand for? Energy requirement. In other words, if a cell has many mitochondria, lots of mitochondria, then what? It has large ATP demands. If it has fewer mitochondria, it has lower ATP demands. Okay? And so the number of mitochondria vary. Like your skeletal muscle are jam-packed with mitochondria. And then contrast that to, say, a fat cell. Not as much, because our fat cells are more for storage, because they're not instead of expenditure. So that depends. And also, the next statement that I have is indicating that within the matrix, you'll also find mitochondrial DNA and mitochondrial RNA. This is unique. This is different than in the nucleus. The nucleus contains DNA for the entire cell, but the mitochondria has the code to make its own transporters and enzymes to carry out its own little processes. Does anybody know where your mitochondrial DNA comes from? Your mother. So your mitochondria are paternal inherited. So whether you're male or female, your mitochondria is identical to your what? Your mother. Who's identical to her mother, which is identical to her mother. So you would also find that in the matrix, keeping the mitochondria doing its thing. What questions do you have about the primary structure of the mitochondria before we move on? We'll look at ribosomes. We'll come back to this, chapter 3. We'll go up pretty good. Oh, let me back up. That will conclude our membrane-bound structures, our membrane-bound organelles. The remaining structures are also found in the cytoplasm, but they lack a membrane around them. They don't fall into our organelle definition. But if you remember our cell landmark, we're looking at the cytoplasm. So you'll find membrane-bound organelles, which we just discussed, as well as ribosomes in the cytoplasm, which are structures important for what? Protein synthesis. We already noted that they may be associated with membranes like the rough ER or the nuclear envelope. They may also be found free in the cytoplasm and still work just fine. I'm going to bump down. We learned that if ribosomes are attached to, say, the rough ER, those proteins that it makes go where? Outside of the cell. Conversely, ribosomes can be floating free in the cytosol, still make proteins, but the proteins won't have what around them? A membrane. So they're considered to be free or dissolved, and they would be functioning where? Within that cell. Cell makes the proteins in the cytosol and uses them to work inside of that cell cytosol, like an enzyme, like the cytoskeletal proteins that we're going to name in just a minute. Does that differentiation make some sense? If a ribosome is associated with the membrane, the protein that it makes is going to go out. Or if a ribosome is not associated with the membrane and it makes a protein, that protein is going to stay inside of the cell. And if you remember from your your basic kind of gen bio, you know that a ribosome only when it's fully complex is it working. So it can join and disassociate. And what happens is it can move from the cytoplasm and then associate to a membrane and then go back to the cytoplasm, whatever. It doesn't, if it's associated with the rough ER, it doesn't have to stay there indefinitely. It go work in the cytoplasm or whatever. Does that make some sense? It's not constrained. Any questions about ribosomes before we look at some other cytoplasm components? Right, so inside of the cytoplasm, we have our membrane-bound organelles. We have ribosomes. We also have something called the cytoskeleton, which is, although it's not bone, it's providing what? Structure to the cell. And what type of molecules are these? Proteins. And it's just a big network of proteins so that our cell can work together. The cell is very complex, and we need to move particles from one spot to the other. And so our cytoskeleton can help in this. There are 3 families of proteins. You ever used the word family before? Family talks about what? Member. Okay, so there's more than one type. Okay, so we have microtubules, we have active microfilaments, and things called intermediate filaments. These are different families. The specifics, whether you're looking at a muscle cell in the human body, in the skeletal muscle, or a protein inside of a fish muscle, might be from the same family but built a little bit different. But collectively, we would call them microtubules, microfilaments, or intermediate filaments. So let's investigate each of these in a little bit better detail. Excuse me. Another just kind of survey of what the cytoskeleton does. Gives physical support. It's involved with transporting particles. I'm going to elaborate on that second bullet. If you remember, we said vesicles are transported from the rough ER to the Golgi to the membrane. It's actually using our cytoskeletal transport system. Well, organelles don't settle down to the bottom of a cell due to gravity because they're attached to the cytoskeleton. If you remember last week, on Wednesday or Thursday, we talked about how proteins can join cells together. They're attached, those membrane proteins are attached to the cytoskeleton to give rigidity and structure and support. We'll probably spend most of our appreciation of the cytoskeleton when we look at muscle contraction, skeletal muscle, smooth muscle, and cardiac muscle. And other cells besides muscles can physically move, like say your white blood cells can move about in the environment. And it's all due to the intricacies of how the cytoskeleton functions. The primary families include what are called microtubules. The micro we can handle, the small, but they form what? Tubes. Okay, so these are the hollow structures. And diameter-wise, these are the largest. Okay, these have the greatest diameter of all the cytoskeletal families. They have that big hollow lumen. They're long and they're hollow. The protein that makes them is appropriately called tubulin. Not too fancy there. Tubulin makes microtubules. Some functions of the microtubules include moving those vesicles like we identified just a moment ago, between organelle to organelle. Very important structure to cilia. What are cilia? They're described as, everybody's doing this. Described as hair-like projections, the little plasma membrane extensions that can move fluid across the cell or move an entire cell within the environment. Also also flagella, described as tail, or just plasma membrane extensions that give locomotion to an entire cell, like in the human spermatozoa. If you take micro, okay, some other microbes have one or two or more flagella. You are probably most familiar with microtubules when you studied cell division and you talked about mitotic spindle and meiotic spindle. Do you remember using those words? And how the chromosomes push and pull? That's quick growth and disassembly of microtubules. They're microtubules that grow and shorten really quickly. And so that's a probably important thing to understand about microtubules is they can elongate really quickly and disassemble really quickly. Because they grow, it's called growing, but just add to them really quickly and then shorten really quickly as well. It's like, you know, remember how long mitosis happens? How long does it take? The actual division itself. Take long? Like 3 days? I don't know how long. It's actually not that long. The actual division itself maybe like 30 minutes. Not very long. So those microtubules extend really quickly and pull back, contract really quickly. Just got a few images to help illustrate the microtubules. I'm going to work right to left. And the image to the right is a transmission electron microscope image of a nerve axon, which we're coming up in Unit 2. Nerve axons, nerves, make vesicles. Can I catch the name of what's in the vesicles? What's in our vesicles? Did you say enzymes? Not enzymes. It's kind of like a hormone, but we don't call it a hormone. It would start with an N. Neurotransmitter. Our vesicles compound, right, so found the vesicles. But those vesicles have to get transported from what we call next week the cell body all the way down the axon and stored at the end of the nerve cell. And so there's actually like a railroad system inside of the axon to make sure the little vesicles get shipped to the right region. And so can you see the tracks here? Those aren't railroad tracks, those are little microtubules that are just serving as a passageway. It's really interesting how it works. I mean, it's busy, right? There's just like a highway. Instead of cars, you have vesicles being transported back and forth along the microtubules. But if you look in the image to the left, we can see that the microtubule assembly is basically these tiny little, use the word globular, is that okay? You see the yellow and the orange blobs? Globular describes the protein as what? Round. It's just a bunch of these round proteins adhered together, forming a what? A tube. And so the whole thing is called a microtubule. We can put little proteins at the end really quickly, or take them off. The plus means add, minus means absorb. You can add them to the end really quickly. This image probably tells a little bit more, right? Makes it a little bit easier for you. The tubulin proteins are the purple, but the whole structure would be called a what? Microtubule. And then here would be a vesicle that's being moved. I could have shown this between the raphia and the Golgi. It's used to traffic vesicles from one spot to the other. Very cool how it works, utilizing these things called motor proteins. You see the green? They actually look like they walk the step in specific directions. This makes me think there's should be, if you go into D12, the very bottom of the content section, there's an area that says supplemental information. Open that link and there's a video, I have it titled Harvard. Watch that Harvard video and this will put it in perspective. how those microtubules are used to transport vesicles. So, under the content section, supplemental information, look for the Harvard video. It's really cool. It shows vesicles moving, etc. So the primary thing about microtubules is they're large and they're hollow and they're used for transporting vesicles and other structures as well. What questions do you have about a microtubule at this point? This is one type of cytoskeletal protein family called a microtubule. We can compare those to microfilaments. On the size scale, these are actually the smallest in diameter. And the protein that makes them is called actin. And you'll become most familiar with these when we get into the muscle component as well. These are contractile proteins. So we'll see these in our skeletal muscles, but also in the digestive system. These help support these things called microvilli, these little cell surface extensions. You can see how they're slender, but they're also made up of what shaped protein adhered together? They're made of globular protein. So we put them all together, they look like a string. So microtubules, diameter-wise, are the greatest. Microfilaments are the thinnest. Okay, so there must be some that are in the middle, and we call those intermediate. I'm not too tricky there. Before I get to the intermediate filaments, you can see this cell. It looks like almost a thing, right? All this is a plasma membrane extension. Proteins, microfilaments pushing this way. Imagine the plasma membrane, they're just pushing out like this. So that when that active filament pushes forward, the plasma membrane needs to push forward. It looks like the cell's walking or something like that. And it's due to the presence of these contractile proteins, microfilaments. And we also identified that the next level of size-wise of our proteins are called intermediate filaments. These, we'll talk about some this semester, but not too much really. But you're probably most familiar with these at the surface of the skin and also the protein in your hair called keratin. That is an intermediate filament. So it gives like physical, tensile structure to a cell. It does describe here like mechanical or physical stress. Is where you'll find a lot of the intermediate filaments. So think about your hair, like how strong it is. It's how it distributes protein, like in your skin too, right? Skin doesn't just break off by holding your pin or something because we've got these big proteins that keep these strong. Collectively, the distribution of cytoskeletal proteins varies between cell types. Like in our skeletal muscles, we'll have more of our microfilaments. In your skin, you'll find more intermediate filaments, etc., etc. So the composition of cytoskeleton varies between cell types. Is an easier way to say that. They may have some or all of these proteins, but the proportion varies from cell type to cell type. The next image that I have shown just kind of helps illustrate the networking ability of the cytoskeleton. These are microscopic, and so sometimes they're difficult to see, especially if you're looking at a light microscope like we'll do in lab today and at the end of the semester. I mean, they're just so tiny, we can't see them with a light microscope. And so sometimes these little kind of artistic renditions help us appreciate how diverse these proteins are and how widespread they are within the cell. So all of these structures will be summarized for you and should be table 2.2. Refer to table 2.2 for a summary of these intracellular structures. multiple cells together. And they're called junctions. They're called cell junctions. Membrane junctions are going to help regulate not only the adhering of cells together, but the joining of different cells together, and then the transport of molecules between cell types. If we look at the primary types of junctions that we have, they include tight junctions, desmosomes, and gap junctions. That's all called desmosomes, or you can call them adherents that we have on them. I'll come back to that. The three primary junctions between cell types that we'll use this semester are called tight junctions, desmosomes, or gap junctions. Two of them are fairly descript, and then one of them not necessarily real descript to you at this point. But if we look at the point of them, this is, say, for example, a cross-section of an organ in the digestive system. We can see multiple cells, epithelial cells, but they've got to be adhered to connective tissue. If they aren't interlaced with proteins, they would be kind of distributed and would be forming a structure, right? And so the tissue would just kind of slough apart. And the way the cells are held together are by utilizing these different types of proteins. Does that make sense? How we use these proteins in the cell membrane? The first type of junction is called a tight junction, and as its name implies, it forms a tight barrier between the plasma membrane of multiple cells. In the images that are shown on the far left, it's just a snapshot of two different cell membranes. So the rest of the cells that are removed, it's just looking how the membranes are held together. The proteins that adhere them together are shown here in the red. And you'll notice the protein of one membrane is interlocked with the protein, same type of protein, with the adjoining cell membrane. They just kind of button up next to one another, if you will. In a tight junction, is there a lot of interstitial fluid? No. And then actually where the junction is, it's kind of a sealed off site. And so do you think it's likely for something to randomly move from one plasma to the other? No. And that's what our tight junctions do. They provide a tight barrier. They prevent any movement from one cytoplasm to the other. It's described as being generally impermeable. Well, hard to say always or never, but it's very unlikely that something is going to simply diffuse from one cytoplasm to the other where there's a tight junction. And so your skin and your epithelial tissue is where you'll find a high proportion of these tight junctions. Tight junctions form a tight barrier, preventing movement of molecules from one cell to the other. The next type of junction that we'll see throughout the semester is called a desmosome. In the desmosome, it's a junction between two cell types. It also looks pretty impermeable, but that's not the primary role of it. Looking at it, what do you think the job of the desmosomes? Distribute what? What are we distributing? It is for distributing. Tension. It distributes tension over a wide area instead of a focalized point. Why might I want to distribute tension? So it can't break. So you kind of keep it together so it doesn't break. So very good. Desmosomes are in tissues that are susceptible to breakage because they're generating a lot of force. Where we're going to see them this semester is in your cardiac muscle. So think about your heart. It is functioning since you're about a 11-day-old embryo. These little cells are generating tension all the way till the bitter end, right? And so they're pumping, and think about when you exercise or whatever, so much force on the heart. We don't want those cells to just randomly rip apart and have a heart attack randomly, right? And so we've got to be able to generate the force in the cells. We want the cells to generate force, but we want that force to become damaging. And so the cell membranes are joined by these things called desmosomes. And if you notice, it's a really complex structure. On the cytoplasmic side, we can see some of our cytoskeleton is attached to a little disc. And that disc has integral membrane proteins attached to it that go out the extracellular fluid, and they interlock. Kind of like that mess that I drew on the board on Thursday. They interlock with proteins that are kind of symmetrical or similar to it. And so, I usually do this. Take your fingers, let's do 2 scenarios. Take your pinky fingers, this will help that. And just do the ends, if you do too much, it won't work. But just do the ends of the pinky fingers together, and then just see if you can break that attachment. Can you break that attachment? Let's do the same thing with your 4 fingertips. Can you break that? So you're generating the same amount of tension, if not more, but if you have more focal points, you can distribute the tension better and it's less likely to break. And that's kind of how these proteins in your finger, in that example, your fingertips are the proteins, and they're distributing it through the attachment. But if we were to really illustrate it, we would intertwine and angle our whole fingers and jumble it up, if you will. And so we're interlocking even more, you can see how strong that kind of junction is. So these are called what? Desmosomes. We'll conclude with the third type of junction, which is called a gap junction, and it forms a gap. Okay. And this time, it's not just a channel out to the extracellular fluid, it forms a channel into the cytoplasm of the next cell. And so what happens? Things flow through. Things like ions. We're going to see this semester for ion transport from one cytoplasm to another. What do ions carry? Charge. We can carry an electrical impulse from one cell to another through gap junction. We're out of time today, so we'll conclude here. I'm going to ask that you read as much as you can in Chapter 3, if at minimum the chapter summary. But we're going to get into metabolism, which is a doozy. So cover as much as you can in advance. Maybe you want to get into the homework number 2. I think I have some videos in homework number 2 that will be relevant to metabolism. So do as much as you can on your own so that Tuesday and Wednesday aren't traumatic for you. We'll conclude here. We'll start lab at 11, or we'll see you in class tomorrow.